to a clean test tube, followed by addition of 2 drops of iodine solution. Observation. For example, you may say blue-black coloration was observed in the test tube containing solution A. Therefore here, column number 2, column 4, procedure, column 4, observation, has been written in past tense. But the last column of inference, simple present tense, is used. Why? Why the last column should be in simple present tense? Why? Think it. The reason behind is that, for instance, you are doing that experiment today in a certain specimen. Let's say our specimen is onion. Then, if you observe that the specimen contains starch today, then even if another scientist may come, let's say after 20 years, still the results will be the same. That is why we are using present tense for the case of inference. But why are we using simple past tense in procedure and observation? Why? Think it. We are using past tense because, first of all, you are required to conduct an experiment. After doing the experiment, you sit down and you write the report. That is why that report should be written using past tense. I think you have understood here. Now, let us go to principle number 2. Principle 2, you should be familiar with the reagents which you are going to use. You should be familiar with the reagents which you are going to use in each test. For example, let's say you want to test the presence of starch in your sample solution. You are required to know the reagent which is used in testing starch. For instance, here you may say iodine solution is needed. Or let's say if you want to test the presence of reducing sugar. You should also know the reagent which is required here. For instance, you may say Benedict solution is needed. If we want to test non-reducing sugar, you should also know the reagents which are needed there. For instance, in that case, you need dilute hydrochloric acid. You also need sodium hydroxide solution. You need Benedict solution. And if you want to test the presence of proteins, you should also know the reagents which are needed there. For example, sodium hydroxide solution. You also need copper 2 sulfate solution. Let's say if you want to test the presence of lipids, the reagent which is needed is Sudan 3 solution. Why is it important to know the reagent which is needed in each test? Why? Think it. Why should you know the reagent needed in each test? Okay, it is important to know the reagents here which are needed in each test in order to perform the correct procedure. For instance, you want to test starch, but instead of using iodine solution as a reagent, you are using, let's say, Benedict solution. Can you get the results there? So if you use Benedict solution for the test for starch, even if starch is there, you cannot get correct results. That is why we are supposed to know each reagent which is used in each test. It is very, very important. That is number 2. Let us now go to principle number 3. Number 3, you are required to be familiar with the color of each reagent. I said that principle number 3, you should be familiar with the color of reagent which you are going to use. For example, the color of dilute hydrochloric acid, that is colorless. The color of sodium hydroxide hydroxide solution, which is also colorless. Color of Benedict solution, blue. Color of copper 2 sulfate solution, which is also blue. color of iodine, some people says that iodine is yellowish in color. Others says it is orange in color. Some says it is brownish in color. But actually, it depends on the amount of iodine in water. The iodine color is brownish. But when the concentration is little in water, you may see as if it has been mixed with the brownish color and yellow, or brownish color and the orange. Therefore, you should be very, very specific knowing the color of each reagent. The question comes, why is it important to know the color of each reagent? Why? Think it. It is important because in writing your report, sometimes you may get a negative result for certain food substances. For instance, let's say you have been tested starch and you observed that starch was absent. Therefore, in observation column there, you are required to write the color which you see in your sample solution. For instance, you may say the sample solution retained brownish color of iodine solution. You cannot say the sample solution retained the color of iodine. You cannot say like that. You should be very, very specific by specifying the color of your reagent. That is why you are required to know the color of each reagent which you are going to use. Let us go to principle number 4. You are required to be familiar with the units of measurements. Why? You cannot say 2 sample solution were poured into a test tube followed by 2 iodine solution. You cannot say like that. You should specify the units. 2 in terms of what? In terms of what? You should be very, very specific. Here, you are required to measure what? Volumes. Therefore, you should know the units for volumes. For example, you may say 2 cubic centimeter. 2 cubic centimeter were poured in a clean test tube, followed by addition of 2 drops of iodine solution. You see? The unit here is drop, while here is cubic centimeter. You should be very, very specific in terms of units. We know the SI unit for volume, it is liter. Liter. But you cannot take a liter of sample solution and pouring in a test tube. You are required to use little amount of it. The little amount of it should be milliliter. Milliliter. But if you read some books, for example, Biological Science Volume 1, have used the cubic centimeter. 1 cubic centimeter is equivalent to 1 milliliter. Therefore, even if you use cubic centimeter, no problem in writing your report. I think you have understood. But some students, they write in this way: 2 square centimeter. 2 square centimeter. Square centimeter, this centimeter, square centimeter, is not the unit for volume. This is the unit for area. Or others, they just say 2 centimeter. When you say 2 centimeter, this centimeter alone is the unit for length. Therefore, you should be very specific to units. You should specify which unit are you going to use for the case of volumes. I think you are clear with the point number 4, familiar with the units of measurements. Apart from that, we are also required to know the principle number 5. Principle number 5 is you should be familiar with the procedure which you are going to use in each test. How you are going to go about. You should be familiar with the procedure of testing each food substance. Procedure for carrying out or for identifying starch, procedure for identifying reducing sugars, procedures for identifying non-reducing sugars, procedures for identifying proteins, etc., etc. You should be familiar with it. Therefore, these are the principles which should be followed in writing report in food testing. After knowing the principles, I said that these are the principles which should be used in writing the food test report. Let us now go to question number 1. Let us go to question number 1. Question number 1 is this one. You have been given specimen A. You have been given specimen A. Prepare a solution from specimen and label it solution A. You have been given with this specimen A. You are required to prepare solution. And after preparing the solution, label it solution A. Clear? Part A of the question. Write the procedure that you have followed to prepare the solution. You are required to write the procedure that you have followed to prepare solution. Clear? Therefore, let's say the specimen which has been given to you is an onion. Let's say you have been given an onion and you are required to prepare solution from it. How should you go about in preparing solution from an onion? How should you go about? Think it. How? Or you can take the whole onion and putting in the test tube. How can you prepare? Okay, here you may prepare your solution First of all, you are required to peel your specimen. You are required to peel your specimen by removing the outer skins. After peeling, you are required to slice. You can say to slice or to cut your specimen into small pieces. Why? In order to be easier for you to grind. After cutting into pieces, you are required to grind. You are required to grind your specimen. After grinding, you are required to add water. After adding water, you are now required to remove solid particles from the solution in order to get a clear solution. And in removing solid particles, care should be taken. Care should be taken. You are not required to filter. You are not required to filter, because when you filter, there is high possibility of removing starch. Therefore, even if starch is there in your sample solution, when you filter, you can remove the starch, and when you test it, you can get negative results even if starch is there. That is why we are not required to filter. But also, decantation is not needed. For the same reason. I think you know the procedure for decantation. First of all, you leave your sample solution there to settle down. After settling down, then you decant. When the solution settles down, all the starch will be on the bottom of your container. And when you decant, you will get the solution with no starch. That is why decantation also is not recommended. You are required to use any other means of removing solid particles there rather than by using by filtration or decantation in order to get your solution. Clear? Okay? Then, after getting your solution there, now you are required to label it. You have already prepared your solution. You are required to label your solution. Label it solution A. A have been provided in the question. Don't use any other letter. Use the letter which have been provided in the question. Clear? From there, after labeling, it means you are now required to write the report concerning the procedure. As usual, the report here should be in past tense. Use simple past tense in writing the report in this part. Don't command. Don't say peel or slice or grind or add water. Don't command. So avoid using commanding language. You are required to write your report using past tense. For example, in procedure 1, you may say specimen A was peeled. Specimen A was peeled and then sliced into small pieces. I think you know the way of using past tense in this part. Then, let's say you are through with writing your report. You have already labeled your solution. Then what next? Obviously, you are required to go to part B of the question. Part B of the question is this one. It says that use the given reagents to identify the food substances present in the solution. That is our part B. But before going to that part, it is very, very important to know the basis of each test. It is very important to know the basis of each testing. For instance, let's say you know the procedure for testing starch. That means, for example, for the case of procedure, you may say 2 cubic centimeter of solution A were poured. You can use the word poured or put. You can say 2 cubic centimeter of solution A were poured or were put in a clean test tube, followed by addition of 2 drops of iodine solution. You know the procedure. You also know the observation. Let's say, observation, blue-black coloration was observed in a test tube containing solution A. Let's say that is your observation. Inference, you know. For instance, you may say starch is present. So you know procedure, you know observations, you also know the inference. But the question is, what is the basis of that test? What is the basis of that test? Here, the basis of test here is that when iodine solution is applied, the iodine reacts with the starch. It reacts with the starch, the iodine solution which is there, reacts with the starch and forms the polyiodide polyiodide complex. Therefore, we expect to get the polyiodide complex, which is blue, black in color. That is the basis for starch. Iodine, which is found in iodine solution, reacts with the starch, which is found in your sample solution. The result of that reaction is the formation of polyiodide complex, which is blue-black in color. Clear? From there, you can now go to another aspect. Now, we are going to reducing sugar. We are going to reducing sugar, the basis of that testing. You know the procedure for testing reducing sugar. You know it. You know that you should have your sample solution and your reagents together with the source of what? Source of heat. For instance, in that case, you may say that 2 cubic centimeter of solution A were poured in a clean test tube. Followed by addition of equal volume of Benedict solution. That is the procedure. You are familiar with it. Equal volume, it means you are referring to this volume. You can say equal volume, or you can specify the volume by saying followed by addition of 2 cubic centimeter of Benedict solution. After adding, you are required to boil. Then the mixture was what? Boiled. The mixture was boiled. You know that. You also know the expected observations. You know the expected observations. Here, you expect 2 things. Whether to get a positive result or to get negative results. If sugar, reducing sugar is there, you can get positive results. If reducing sugar is not there, then obviously you can get what? Negative results. For instance, let's say in an observation you observed the series of color change. A series of color change from the color of reagent, that is blue, from blue to green to yellow to orange up to brick-ready precipitation. Let's say you have observed the series of color changes there. You know the way of boiling. You know you are not required to boil continuously until reaching brick-ready. When you boil continuously, obviously you cannot see the series of color change. You can see only blue color and the brick-ready coloration. You are required to boil for a while, then you remove it. You boil again, you remove. You boil, you remove. By doing that, you can see the series of color change. I think you know it. But my intention is not that. My intention is not that to introduce to you on how to conduct that procedure, because you know. I'm interested with the basis of that test. I'm interested on this part. Therefore here, I said you expect to get the series of color change which ends to brick-ready coloration. It depends on the concentration of reducing sugar which is found in your sample solution. If reducing sugar is little, you cannot get brick-ready coloration. Therefore, as concentration of reducing sugar increases, you also increase the chance of getting brick-ready. Clear? What is the basis of that test? You know that reducing sugar, you are using Benedict solution. Benedict solution, one component of Benedict solution is copper 2 ion. It is made up of copper 2 ion. Therefore, when you apply Benedict in your sample solution which contains reducing sugar, then reducing sugar reduces copper 2 ions into copper 1 ion. Copper 2 ions, blue in color. Copper 1, brick-ready in color. And this one is brick in color, brick-ready. But I said that it depends on the concentration. Sometimes you may get the intermediate sort colors. So why are you getting the intermediate colors there? You know the function of reducing sugar. Let's say you have your copper 2 ions, but you have little reducing sugar. When reducing sugar is little, it means some copper 2 ions will be reduced to copper 1 ions. And some copper 2 ions will remain in your solution without being what? Reduced. So you will have what? Mixture of copper 2 ions and the copper 1 ions in your solution. This mixture makes you to get the intermediate colors. You may get either yellow or orange depending on the concentration of it. So that is the essence behind. Clear? But the copper 1 ion which is formed here is the copper 1 oxide. It is copper 1 oxide. Therefore, from copper 2 sulfate, which is found in Benedict solution, is reduced into copper 1 oxide. Clear? It is my hope that you have understood that concept. So, the essence is that copper 2 ions reduced to copper 1 oxide, which is brick ready in color. You get the intermediate color. I have already explained the reason behind. Clear? I think you have understood there. Okay. Now, before going to explain how will we use the reagents, you should know that. You should know all the bases in each test. For this time, I managed to discuss with you only 2 bases. Bases for starch and bases for reducing sugar. Next presentation, I will continue from where I ended. I will continue with the non-reducing, then protein up to lipids. Clear? Therefore, let me end my presentation here. I invite you to my next presentation. Don't miss. It will be very, very interesting. Therefore, I beg you, join me in another presentation. Thank you so much.
Uongozi wa Chuo Kikuu cha Mtakatifu Agustino Tanzania, tawi la Mbeya, unapenda kuwakaribisha wahitimu wote wenye sifa za kujiunga katika kozi mbalimbali zitolewazo na chuo. Chuo kinatoa kozi ya shahada ya juu ya elimu, yani postgraduate diploma in education, shahada ya kozi ya elimu, yani Bachelor of Arts with Education, shahada ya utawala wa biashara, Bachelor of Business Administration, na katika michepuo ya rasilimali watu, Human Resource, ugavi na manunuzi, Procurement and Supply Chain Management, usimamizi wa masoko, Marketing Management, uhasibu na fedha, Accounting and Finance, benki na fedha, Banking and Finance. Vile chuo kinatoa stashahada, diploma na stashahada certificate kwa kozi za utawala wa biashara, Business Administration, sheria, law, teknolojia ya habari na mawasiliano, ICT, uhasibu na fedha, Accounting and Finance, ugavi na manunuzi, Procurement and Supply Chain Management, usimamizi wa masoko, Marketing Management, rasilimali watu, Human Resource, ujasiriamali, Entrepreneurship Development, maendeleo ya jamii, Community Development, ukutubi, sayansi, taarifa na tehama, Library and Information Studies with ICT. Kwa mawasiliano zaidi, piga namba zifuatazo: 0684797866 au 0764622482 au 0025525004240 au tembelea tovuti ya www.sautimbeya.ac.tz.
Kipindi cha Darasa Online kimeletwa kwenu na Chuo Kikuu cha Sauti Mbeya.
Katika kipindi cha Mizani wiki hii, tutakuwa na mkurugenzi wa jiji la Dodoma Godwin Kunambi, tukijadili kwa kina maendeleo ya ujenzi wa makao makuu mapya ya mji wa Dodoma. Usikose kipindi hiki ambacho kitaongozwa na Dr. Ayubu Rioba, mkurugenzi mkuu wa TBC. Ni Jumatano tarehe 3 mwezi Juni mwaka 2020 saa 3 usiku, na marudio yake ni Alhamis saa 9 alasiri hapa TBC1, penye ukweli na uhakika.
pongezi nyingi twatoa kwa rais wetu.